In looking back at how substances moved across the plasma membrane, we have just seen that some can move passively by passive transport mechanisms like diffusion and osmosis. But the cell can also make use of active transport in certain situations when moving substances across the membrane. However, unlike passive transport, active transport requires cellular energy input from the cell, and this energy typically comes in the form of ATP. So active transport typically makes use of protein pumps that are powered by ATP, and these protein pumps of active transport are typically moving substances from areas where that substance is low in concentration to areas where that substance is in high concentration. And so this is the opposite of diffusion and osmosis, and this is also why active transport requires energy, because we're moving substances against a concentration gradient, that is, we're moving them from low to high. Whereas with diffusion and osmosis, we saw movement from high to low. So in order to be able to move these substances against concentration gradients, the active transport mechanisms need ATP energy to fuel them or to drive them. So when cells actually use ATP for energy, the cell breaks the chemical bonds that are holding the leading phosphate group here onto the other phosphate group. So it breaks approximately this bond right here. And chemistry tells us that when chemical bonds are broken, they tend to release energy. And so because this energy releasing, this bond breaking process occurs in the presence of water, we refer to the breaking of ATP as ATP hydrolysis. So since ATP is being broken to release energy in the presence of a watery environment. So that's ATP hydrolysis and how we release energy from ATP. So let's turn our attention back to the pump mechanism for a second here. So how exactly does the pump mechanism work? So before ATP actually binds to a pump, the pump is in a sort of inactive configuration, if you will. And when it is in that inactive configuration, it does not move substances across the membrane. So that is before ATP is bound to it and before ATP has been hydrolyzed, the pump will not do any work for the cell. So once ATP is actually bound to it, ATP is hydrolyzed and split, and this leading phosphate group is split from it. This releases energy. So as I mentioned earlier, basic chemistry tells us that breaking chemical bonds releases energy. And from this release of energy, what we get is the activation of the pump mechanism. And when that pump is activated, it moves that specific substance that it is designed or designated to move. So meanwhile, ATP has lost its leading phosphate group, becoming a molecule called ADP, which stands for adenosine diphosphate. So there's 2 phosphate groups on this molecule that remains instead of 3 phosphate groups. So the phosphate group that was broken off of it will remain on this pump for a short time, after which it is ejected from the pump, and then a new ATP molecule can bind to it and begin the process again of pumping that substance across the membrane. So there are 2 types of active transport. We have primary active transport and secondary active transport. And the reason that these 2 differ, not only in their name, but they also differ slightly in their function, we see that primary active transport actually makes use of that ATP hydrolysis energy directly, and secondary active transport actually uses ATP energy, but in an indirect way. So let's discuss primary active transport, and then we'll take a look at secondary active transport to see how that works as well. So beginning with primary active transport, so this diagram is actually showing both transport mechanisms. On the left, we have primary active transport here, represented by this purple pump mechanism here. And then on the right, we have secondary active transport mechanisms shown by these little magenta transporter proteins here. So on the left is this pump that engages in primary active transport. It's very appropriately called the sodium-potassium pump, because you can probably guess what it pumps. It moves sodium and potassium across the plasma membrane, and it does so using ATP energy. So let's take a look at how this works, right? So this is kind of a time lapse, if you will, of the the mechanism of the sodium-potassium pump from left to right here. So we have our sodium-potassium pump, and we have our plasma membrane here. This yellow down here, this yellow region down here represents intracellular, so this is inside of our cell. The blue environment up above it represents the exterior or external environment, or extracellular environment. So the sodium-potassium pump begins by binding 3 sodium ions from inside of the cell and a single molecule of ATP. So that ATP in the next step is then split or hydrolyzed. That releases the energy, and you can see that the pump here changes shape and changes configuration so that it opens up the pump and ejects these 3 sodium ions that it has bound. And so it's kicking 3 sodium ions out of our cell into the extracellular space. And at that same time there too, as it's ejecting those sodium ions, it's actually picking up 2 potassium ions. And these 2 potassium ions bind to a very specific and different site on the protein pump than the sodium ions do. And so those potassium ions then bind, and then the pump reverts back to its resting shape or state when it loses that phosphate group that was cleaved or chopped off of ATP. That changes its shape back to its original form, and it releases the potassium into the intracellular compartment or fluid there. And from step 4 here, we're now able to go back to step 1 if another ATP comes along, as well as some sodium that bind to it as well. So that's a little bit of the general mechanism of primary active transport. So let's look at the functions of primary active transport. So overall, what is this accomplishing in the cell? So for one, the primary active transport maintains cell volume because water tends to follow the movement of both sodium and potassium. So wherever both sodium and potassium are going, water tends to follow passively by osmosis. Next, it also powers secondary active transport. So secondary active transport is highly dependent on the action of primary active transport, and we'll see that here in a few minutes. Primary active transport also maintains a state or a condition in the cell that's known as resting membrane potential. So let's dive into resting membrane potential and take a look at what resting membrane potential is. So resting membrane potential, it turns out, is a characteristic that the cell adopts when it is in its unstimulated state. So by using the term resting membrane potential, we're implying that the cell is in an unstimulated state. And so resting membrane potential is actually a voltage difference. So you could easily interchange the term potential with voltage. So if you said resting membrane potential or resting membrane voltage, then those two phrases would be equal to one another. So those are equivalent to one another. And really all a voltage difference is a separation of electrical charge across our plasma membrane here. So in this way, a cell kind of behaves like a little battery, because batteries behave in that way too. They operate by first establishing or by using the separation of charge to create electrical flow or current. And so we see the same thing in cells. We see that there's a separation of positive and negative charge across the plasma membrane. So what we actually end up seeing is that the interior of the membrane is slightly negatively charged at about minus 70 millivolts, which is about minus 0.07 volts. So it's a really low negative voltage, but it's still a negative voltage nonetheless. The inside of the plasma membrane is slightly negatively charged with respect to the outside, or compared to the outside of the plasma membrane. And it turns out that this separation of charge is created by the differences in concentration of sodium and potassium across the membrane, as well as the ability of these two substances to diffuse into or out of the cell. So what we see is that at rest, there is a higher concentration of sodium outside of the cell, which is represented by these purple positively charged ions here. So there's a higher concentration of sodium outside of the cell than we see inside. And this difference causes sodium to diffuse into the cell. And so sodium has a tendency to want to diffuse with its concentration gradient into the cell. And in doing so, it tries to raise the voltage of the plasma membrane, but it's unsuccessful, and I'll say why it's unsuccessful here in a little bit. So we see that sodium diffuses in, and it actually does raise. Maybe I should go back and say that it does raise the membrane voltage of the plasma membrane a little bit, but the fact that potassium is actually leaving the cell at a greater rate than sodium is entering is the reason why we get this negative resting membrane voltage here. So returning to potassium here, we see that potassium in a cell at rest is generally at higher concentrations inside the cell than outside, and so potassium is represented by these little orange ions with a plus icon or with the plus symbol on them. And so we see that there are higher concentrations of potassium inside the cell than outside, which causes potassium to diffuse out. Now, for reasons that we'll go into detail in a little bit later chapters, so in particular the muscle physiology and the nervous system physiology chapters, we see that more potassium actually diffuses out of the cell than sodium diffuses in, because both sodium and potassium are positively charged substances. But what we end up seeing is that more potassium ends up diffusing out per unit time than sodium does diffusing in. And so because we have more positive charge going out than we have coming in, our membrane voltage reads slightly negative, right? Because a negative resting membrane voltage means that there's more positive charge moving out than in, right? So our resting membrane voltage will read as slightly negative on the inside because of the diffusion of potassium out of our cell. And so to permit this continued diffusion of potassium out of the cell and sodium into the cell, the sodium-potassium pump uses ATP to establish these sodium-potassium gradients that we see on both sides of the plasma membrane. Okay, so that's primary active transport. Let's briefly turn our attention here to secondary active transport. As I mentioned earlier, secondary active transport uses ATP energy indirectly. So what we mean by this is that the source of energy for secondary active transport is actually created by primary active transport. And so because of this, secondary active transport is highly dependent on the activity of primary active transport. And what secondary active transport tends to do is it couples the movement down or with the concentration gradient. It couples that with movement up or against a concentration gradient. And so if that statement was a little bit confusing, let's take a minute and unpack that, and I'll show you exactly what I meant by that. So back to our diagram here of our primary and secondary active transport mechanisms. Again, on the left here, we have our sodium-potassium pump, which is functioning in primary active transport. And we saw that the sodium-potassium pump establishes this sodium gradient on the outside of the cell. It also establishes the potassium gradient on the inside of the cell. But in this example here, we'll just ignore the potassium gradient for right now. We'll just focus on the sodium gradient right now, or at this time. And because there is a higher concentration of sodium outside the cell than inside the cell, sodium wants to diffuse back into the cell. And it can do this through the leak channels, through the facilitated diffusion channels that we saw earlier in the chapter. But sodium can also diffuse into the cell through our secondary active transporter mechanisms over here on the right. And what we're seeing here is that these two magenta transport channels here are simultaneously moving sodium and glucose into the cell. So because sodium is at a higher concentration outside the cell than inside, sodium is diffusing down or with its concentration gradient. That is, it's moving from an area where sodium is highly concentrated to an area where sodium is less highly concentrated. And what we see here is that glucose is actually moving against its concentration gradient. So there is actually lower amounts of glucose outside of the cell than there is inside. And so glucose is actually being pulled in by the action of sodium as well. So both sodium and glucose are being transported at the same time. And because sodium and glucose are being transported at the same time in the same direction, these are referred to as sodium-glucose symports, because both of these substances are moving in the same direction across the plasma membrane. So glucose isn't the only substance that's transported by primary active transport. We also see things like amino acids and many ions that are actually moved by secondary active transport mechanisms. So to wrap up secondary active transport mechanisms, I will leave you with these two definitions here, these two descriptions here. So the secondary active transport proteins that move substances in the same direction as we just saw in the previous example are referred to as symports or symporters, because both of these substances are being transported in the same direction across our plasma membrane. Now, certain secondary active transport proteins actually move two substances in opposite directions across the plasma membrane, and secondary active transporter proteins that move substances in opposite directions like this are referred to as antiport or antiporter mechanisms.